My name's Ryan. I'm an honours candidate here at the University of Adelaide, and my research is looking at the timing of neuroinflammation following a spinal cord injury. So a spinal cord injury is a devastating event that causes severe motor, sensory and autonomic functional loss. So just a little bit of a background. There are currently over 15,000 Australians living with the effects of a spinal cord injury, and this is most commonly due to motor vehicle accidents. It's estimated that a single incident of paraplegia costs the healthcare system over $5 million, and this is because there's no cure for spinal cord injury. So the majority of people who have a spinal cord injury are left with lifelong deficits and need substantial ongoing care. So when we're talking about a spinal cord injury, we're really talking about two different types of injury: the primary injury and then the delayed secondary injury. So this primary injury is the initial injury to the spinal cord that's a direct consequence of a traumatic event itself. So say you have a motor vehicle accident and you get a dislocation or a fracture of a vertebrae, which damages your spinal cord tissue. This is your primary injury. And unfortunately, it happens pretty much instantaneously. So once you have a traumatic event, this is immediately happening, which means we've got no therapeutic window where we can actually go in and give an intervention and treat this. But what's interesting is that this lesion, so this area of damaged tissue, doesn't just stay still, but over the next days, weeks and months, it actually expands and covers more of your spinal cord injury. So this is your secondary injury, this expansion of this lesion over time. So now you have more of your spinal cord that's compromised, less and less communication that's able to go through, and worse patient outcomes. This has become a major focus for our current spinal cord injury research because it occurs over a delayed time frame. So we have a potential therapeutic window where we might be able to, if we understand the mechanisms, go in and treat this and make this better for the patient. So there's a lot of research going into this, and we've got we've identified lots of factors for our play during this window of time. But one of the key driving forces behind this is neuroinflammation. Neuroinflammation sounds a little bit fancy, but basically it's just this inflammatory response within your spinal cord after injury. So just like an injury to any other part of your body, your spinal cord goes through this inflammatory, swelling sort of reaction. And we're beginning to realise that there are both beneficial and detrimental aspects to this. The best way to think of neuroinflammation is a lot like the footy teams in Adelaide. It's both very good and very bad. So you have good aspects of neuroinflammation, like the clearance of debris, which allows us to facilitate repair and regeneration. But we also have these bad aspects. So this neuroinflammatory response damages adjacent cells that weren't initially damaged, which expands this lesion. So just like the crows, neuroinflammation isn't the best at hitting the target. There's still a lot of unknowns about this response though, so we really don't know what mechanisms are driving this. And we still don't know really when we're having this more beneficial aspect and when we're having the more detrimental aspects. So this is the main focus for my research. My research is trying to track the expression of inflammatory markers within the blood across the first 2 weeks following a spinal cord injury. So we take a baseline blood sample before a mild or moderate spinal cord injury, and then we take a series of blood samples across the next 2 weeks at various time points, which then get centrifuged, we collect the serum layer, and then analyse this for inflammatory markers within our lab. And what we're expecting to see is a really big peak within the first 24 hours of these more detrimental markers, and a more delayed, closer to 7 days peak in these more beneficial anti-inflammatory markers. And we're expecting this to occur in a severity-dependent manner. So our moderate spinal cord injuries will have a greater expression of these inflammatory markers than our mild injury. So why are we doing this? Our first aim of our research is really to improve our understanding of this neuroinflammatory response. So we're characterising how it changes across time and changes across severity. This leads our research into potentially identifying biomarkers of spinal cord injury severity and progression. So we might be able to say at a certain time point a patient might have this level of marker within their blood, which correlates to a more severe injury. This would allow us to improve patient prognosis and better inform clinical treatment management strategies. Our final goal is really to aid the development of treatments. So we might identify time points that are more suitable to certain interventions, which will allow us to give treatments more effectively where it's expected to have the best effect. Yeah. So we're hard at work in the lab currently, sort of trying to analyse our samples and get some results, so stay tuned for that. But in the meantime, I'd like to thank a huge team of people that's taken to get this project as far as it's got so far. Thank you to the NRF for their support of our lab, and thank you all for listening.